Most women will encounter human papillomavirus, or HPV, at some point in their life. HPV is the most common sexually transmitted infection worldwide and is transmitted via skin-to-skin genital contact. Some cases of HPV clear up on their own. However, others can become a chronic infection leading to cervical cancer. But don't be worried, because cervical cancer is very preventable via vaccines, HPV screening, and treatment of precancerous cells. There are two HPV screening tests. Both tests collect cell samples from the lining of the cervix. From there, the Papa-Nicolau test, or Pap smear test, involves looking for the presence of abnormal cells, whereas the HPV DNA test involves looking for the presence of HPV DNA. Sadly, many disadvantaged women from low socioeconomic countries are much more likely to develop cervical cancer. In an effort to increase HPV screening among these women, self-collected HPV DNA tests have been a focus for researchers over the years. This strategy aims to reduce barriers such as cultural privacy and access to healthcare, which usually prevent these women from undergoing HPV screening. One particular study, based on low socioeconomic women in Mexico, aimed to compare the self-collected HPV DNA test against the Pap smear test. The participants were split into groups, one group receiving the self-collected HPV DNA kit, and the other were directed to undertake a Pap test at a local clinic. Any women who tested positive for HPV were referred to have a colposcopy, a procedure that involves a doctor using a microscope to look at a woman's cervix for abnormalities. There were a number of important findings. Overall, many more cases of HPV and cervical cancers were found among the self-collecting HPV group compared to the Pap smear group. Therefore, the HPV test was much better at picking up the presence of HPV or cervical cancer, meaning the test had a greater relative sensitivity. On the other hand, a positive result from a HPV test was less likely to lead to a confirmed diagnosis of HPV or cervical cancer compared to a Pap smear test. Therefore, the Pap smear test was found to have a greater positive predictor value. Due to the high sensitivity of self-collected HPV tests, it would be helpful to implement them in low socioeconomic countries as they increase the chance of HPV and cervical cancer being detected, which is important as HPV testing doesn't happen many times in these women's lives. HPV self-testing also provides better privacy for women, leading to increased cultural and religious safety. On the other hand, the low positive predictor value for the HPV tests needs to be addressed, as many women received false positive results, which led to unnecessary colposcopies and biopsies. If implemented, this would create strain on public health services and might deter women from completing all of the necessary tests to confirm their diagnosis, due to cost and other personal barriers. If these limitations are addressed, introducing HPV self-testing would be a practical measure to reduce the high rates of HPV and cervical cancer globally, particularly in low socioeconomic countries.